everybody. So I hope everyone has a good first week of the REU so far. So I'm going to talk about the AI applications in bioinformatics and precision oncology. So it includes 4 parts. So I'm going to give some background on molecular biology, which I don't think this group of people had. And then I'm going to touch upon briefly AI and precision medicine. And then I will get into a few examples of my own research, just to show you how AI can be applied in bioinformatics and precision medicine. Okay, so I'll conclude with some of the outlooks. So let's get started here. So first of all, let's take a look at some of the backgrounds on molecular biology. So the first question I will ask is, why are we different? We are actually very similar. We are all humans. We can easily tell us apart from other animals, yet we are also very different. Okay, so the reason that we are so similar and we're also very different it's really because of the DNAs. So the DNAs are long chains of amino nucleotides. So there are 4 types of nucleotides. There are A, C, T, G, right? So DNA, as you can see here, it resides in the nucleus of the cell. So this is the structure of the mammalian cell, where you have a nucleus, and inside the nucleus you have DNAs. Okay, so this is how DNA is really packed. So just briefly talk about DNA. So as I just mentioned, DNA actually are in a 3-dimensional structure are the so-called twisted pairs. So there are 2 long chains of these nucleotides, ATCG. So there are 4 types. These are ATCG are the 4 types of nucleotides, and these every single chain you can consider this as a sequence of these nucleotides, and the 2 chains are bind together through the so-called base pairing, where C always paired together with G and A always paired together with T. So therefore, if you know the sequence order, these order of the letters in one chain of the DNA, you know the order of the sequence of the other chain. Okay, so in fact, the order of these letters in a sequence is very, very important, because it's considered as a so-called genetic code. It codes how an organism should produce proteins. So therefore, these orders of the letters are also considered as a genetic code. So however, well, first of all, in human, we have 3 billion letters. So this is DNA is a very, very long chain, consider many of these nucleotides. However, it's not any parts of the DNA are considered as storing genetic informations. Only portions of the DNA, parts of the DNA store the so-called genetic informations. These parts of the DNA are the so-called genes. Okay? So the genetic information, or these orders of the sequences inside the gene, codes the specific type of protein that the organism or human will produce. Okay, so in human, we have about 25,000 these segments of genes that codes more than 70,000 proteins. Okay, so the fact, now coming back to the question why we are similar or different, the fact that we are similar is because this genetic information that codes in human DNA in you and me are very, very similar. Yet we're also very different. One of the key reasons is there are also subtle differences in the genetic codes between you and me. So that make us different in our height, in our characteristics, in how we look. And if our genetic information is mutated, and that will impact the type of proteins that will produce, and eventually that could lead to disease. So a key part of understanding disease and finding effective therapy is to understand our genetic information and see where exactly in our DNA mutation happens, and to determine the type of mutation that are disease-related, and then find cures to possibly prevent such mutation from happening or reverse such mutation. Okay, so that's why we or I myself study this type of information. All right. However, another reason why we different is also because the process going from this genetic codes to the type of proteins that it produces. So it's really because this process could be also different. And this process of going from DNA to protein is referred to as central dogma of molecular biology. So it involves two steps, namely transcription and translation. So well, let me give you a bigger picture here. So imagine now we just talk about DNA resides inside the nucleus of the cell. However, the so-called protein factory or ribosome that produce proteins sits outside the nucleus. So in order for the ribosome to receive genetic information to know the type of protein that it will produce, the genetic codes stored in DNA inside the nucleus needs to be transferred outside the nucleus to the ribosome. And this process involves another molecule called messenger RNA. So this is the type of messenger RNA that you see. So the messenger RNA duplicates the parts of the genetic information, specifically the segments of the genes, the DNA sequences in the segment of genes. It copies that segment, essentially copies genetic codes, and then and going outside the nucleus and go, so let me back up, and go to the ribosome. Okay, so the process of copying the genetic code from DNA to messenger RNA is referred to as transcription, and the number of copies of mRNAs is often referred to as gene expression. So we constantly produce messenger RNAs or making copies of these messenger RNAs. And the gene expressions or the amount of mRNAs are produced is highly regulated and it can vary from time to time, from day to day. But this process is tightly regulated through very sophisticated mechanisms. Okay. So, and then the second process of taking the codes copied from mRNA and then produce proteins in ribosome is referred to as translation. Okay. Coming back to the central dogma of molecular biology, so I just pretty much described the central dogma of molecular biology. So that describes how protein is produced based on the code stored in DNA. So, and that involves these two processes of transcription and translation. In addition, DNA can duplicate itself. This is when cell duplicates itself, and then DNA will also duplicate. And the amount of mRNA and the protein are referred to its gene expression. This is a very important concept that I will refer to in the applications in this talk. Okay, so as I just mentioned, that this process of transcription and translation is tightly regulated, and even though this regulation is fairly stable in general, however, it also varies based on a lot of factors, a lot of outside and endogenous conditions. So the variation of the conditions renders different amount of gene expression produced, therefore proteins. Okay? And that also leads to changes in our so-called phenotypes, the state of our human, our state of your person, your modes, your emotion, your health, and so on and so forth. So that's another aspect why we different. Okay, even for us, we different in days, right? We different by days. So our health changes. So another one of the key reason is because of this tight regulation of the transcription and translation. Okay, so. And there are many aspects that controls this this process of the transcription and translation. I listed a couple of key factors, and overall, this is a highly sophisticated process. However, understanding the mechanism behind this process is key for us to know the biology of transcription and translation, and then finding new therapies and the cures for disease, and finding ways to possibly improve our health. So researchers in this area put a lot of emphasis in understanding every aspect of genetic regulation of the transcription and translation. And that's one of the key goal of also my research. However, I do not really approach this problem from a traditional biology angle, or in the sense that I don't do bench experiments, the type of experiment you normally see in a biological lab. I approach this research using computation and mostly using machine learning and AI. So therefore, let me quickly touch upon some of the backgrounds on AI. I guess you have already listened to a couple of lectures last week on AI. So let me quickly touch upon that, but mainly focus on its relationship with biomedical research. So first of all, why do we do biomedical research, right? I believe by now you already know from my first section of the talk that understanding the biology is key for improve human health and finding cures for diseases. And it's just let me back up. It's really as you can see, it's really sorry about this. It's too sensitive. It's really a national and international research focus because of those reasons. And you see some of the initiatives. These are national initiatives, including Brain Initiative, Precision Medicine Initiative, and Cancer Moonshot Initiative. These are some of the big projects that are organized by U.S. government. So it really speaks for the significance of the biomedical research. It is also the industrial trends is because human health and healthcare is really one of the largest industry in U.S. and in the world. So therefore, coming up with new therapeutic strategies or new ways to improve health, it's a really profitable market. So a lot of these big companies put a lot of emphasis in healthcare, and you notice that some of the big names that I I noted here are not really healthcare companies. So they are data companies, they're AI companies, including IBM, Google, and Apple. They play a significant role in this market. So, so the question here is, you might wonder, can we really do the so-called apply AI in biology or do so-called intelligent biological research, right? So let me touch upon how we can do this type of research, or what does this type of research really look like, right? So in integration of AI and biological research, now has already ushered us into a new research area. So now we refer this as intelligent biological research, where in this research, AI functions as an intelligent assistant to work together with experts to accelerate research cycles of experimental design, data analysis, and hypothesis generation. So to achieve this, we need to really equip this AI agent with the type of intelligence to be able to communicate, to learn, and to predict, to understand, reason, and lastly, like a human, to be creative, to create a new approach, and so on and so forth, right? So so this is really, we're really at the very early stage of this research, but you know, I, like many of my colleagues in this area, are pretty optimistic that we eventually will be able to build such agents. So what I'm showing you here isn't just the outlook, right? So, but now we're really just at the beginning of building such a AI system. So where are we yet, right? So currently, the so-called AI algorithm at the best can only learn and predict the data. So they don't process the rest of the capabilities that such an agent should process. So there's also a little sign of a current AI model to be able to really perform other type of research tasks yet. So there's still really a long way to be able to build such an AI agent. Okay. However, the research in this area is yeah, just sorry, give me one sec. Seems to have some give me one sec. Sorry, I'm back here. So there's some technical issues. All right. So, well, as I said, we're still at the very early stage. However, there's really a so
Yufei,
AI. Yes. Mm-hmm.
the screen is like a white page.
Right. Yes, okay. I'm moving on to show more here. So, yeah, what I want to say is this is really a very active area of research. So there's a significant increase in the number of publications in high-profile venues about AI and biomedical research. So as you can see, I'm showing you a few examples here. So that includes, let me see. Okay, yeah, I'm showing you some of the example of these high-profile publications of applying AI or deep neural networks in functional prediction of non-coding sequences, the so-called variant calling, and single-cell analysis, in applications of AI in sorry, my computer is really slow. Doesn't, somehow got stuck. Oh, there we go. Okay, so in very calling applications in radiology, in pathology, dermatology, and for predicting clinical outcomes, drug selection, design, and improving healthcare in general. Okay, so here is a non-exhaustive list. Actually, it's a pretty limited list of these AI applications in biomedical research. So it covers many aspects that you as outsider probably heard of, right? So and it really permeates in every area of the biomedical research in this days. Okay, so now oops, it's too fast. Now in medicine, we have envisioned AI to function as eventually to function as a virtual medical coach to provide individualized health recommendations across human lifespan. Okay, so why suddenly AI? So let me talk a little bit about AI now. So why AI? So so this trend it really started about 10 years ago. Okay, and one of the key reasons of there are actually two factors. One of the key factors is really because of the so-called deep learning revolution. Okay, so it jumped into the public attention due to its ability to provide human-level superhuman performances. Okay, initially in computer vision, in speech recognition, and more recently in many different areas that you also heard of. For example, playing chess, in playing video games, and generating human images. As a matter of fact, these two pictures of the human are not pictures of a real human on Earth, right? They're AI generated images. Okay, so yeah. So you probably also heard on the news and this AlphaFold. So it claims that it solves this age-old biological question using AI to really being able to predict how protein folds. And these are a few examples of AI solving big problems, right? So this is one of the factors why AI suddenly become so popular. And another reason is really because of a big data revolution. It's really because of the data. So deep learning is extremely data hungry. Okay, so all these tremendous performances of deep learning that I showed you in the previous slides are achieved by training on huge amount of data samples. Okay, so in biomedical research, advances in high throughput biomedical equipment, such as high throughput sequencing, which I'll show you in the next slides, ubiquitous health-related wearable devices, and then the concerted effort by hospitals and the federal agencies to collect electronic health records, genomics, brain genomics, brain imaging, and mobile health data have provided a fertile ground for applying deep learning. Yeah. So these two factors together propel the current AI biomedical research. Okay, so myself works in this genomics domain. So I want to show you a little bit more about these genomics. First of all, there are many big efforts, big projects that focus on collecting data. So which I listed some of them here. That includes, for example, Human Genome Project, so that sequence the entire this genetic code, or the nucleotide sequences in human And then 1,000 genome project, so that catalog the mutations. And so essentially sequence the specific genome of a human, and then being able to catalog the mutations. So there are also the Cancer Genome Atlas project that produce measurement of gene expression and many other aspects of the regulatory elements profile of this genome-wide regulatory elements of human cancer from real human cancer patients. And these projects produce many of these projects. The sole goal is to really generate data and then release to the public. So these projects really help us provide us with a large amount of data by which we are able to now perform AI projects, AI research. Okay, so I touch upon the so-called sequencing machine. So here are some of the examples of a sequencing machine. So the so-called sequencing machine or high throughput sequencing machine, its goal is to read the specific nucleotide orders of sequence. So for example, if we take a sample from you and then submit it to this machine, the machine will be able to output the specific type of the specific nucleotide sequences of you. So, and the throughput of this type of machine is really enormous. So, and here's one of the examples where it produced about 2.5 billion sequence reads in 3 days. Okay, so and each reads is about up to 150 base pairs. So you can multiply these two numbers. So human genome, I just explained to you, is about 3 billion base pairs. So you can see here, this machine is able to produce about 100. It is able to sequence 100 human genomes in about 3 days and produce about 1 terabyte data. So in 3 days. So here at UT House, so there's one such machine at UT House. The throughput is really huge. So one machine is enough to really service the needs of the entire university. Okay, in San Antonio, I'm not sure, there are probably 3, 4, or maybe more such type of machine. But the machine so has been also tailored or modified. Well, I wouldn't say modify the machine. The protocol has been also modified to be able to not only sequence just the nucleotide content of the DNA, but many regulatory elements in DNA that regulates transcription. So this is a highly powerful machine that's been used in all aspects of molecular biology to collect a large amount of data. So now, well, this is pretty much what I mean. So that DNA this is high throughput sequencing machine has been used to sequence the entire genome, to detect the mutations, or we call variants in the genome, to sequence transcriptome or the so-called gene expression, as I mentioned. So they essentially sequence these mRNAs and then being able to count the amount of mRNAs that's been produced by DNA, and then sequence many regulatory elements. So these are some of the commonly used protocols that this high throughput sequencing machine is being subjected to on now almost on a daily basis, and so using it's too fast. Okay, so using these high throughput sequencing data, and then we apply deep learning and AI, and we're able to be able to really understand how the mechanism behind the mechanism by which human cells really regulates this process of transcription and translation, and then being able to design drugs, but to be able to also understand disease, okay, to be able to tell tell apart for example, the cancerous cells from the normal cells, and then being able to design drugs to provide new therapeutic targets and new type of a drug. So these are some of the common research topics that we focus on, including, for example, cancer diagnosis, predicting, for example, the metastatic tumors, and prioritize drugs, predicting, for example, the drug responses to a specific patient, and then understands the genomic mechanisms, or predicting, for example, the type of genes that a cancer has a so-called cancer dependencies. Okay, so right now, this area, the so-called precision oncology, is a particular type of research and clinical practice that emphasize specifically on the precision at the so-called molecular level, and specifically the goal of precision oncology is to, through molecular profiling, using mostly high-throughput sequencing data, to develop a precise understanding of the tumor development and its microenvironment, the diversity and the treatment response in individual patients, among populations, and over time, so that diagnostic and prognostic markers and targetable alterations and the mutations can be identified for effective targeted therapies and immune therapies. And AI plays a significant role in many aspects of this research, including formulating questions, design experiments, interrogate and interpret data, reasoning and provide decisions, and then generate hypotheses, identify targets. So this is, as I said, it's a very active research area. Okay, so now let me see, what time do we have? 9:40. Okay, let me use the remaining 20 minutes to show you some of the examples of how AI can be applied in this area. Okay, but before I get into these examples, let me quickly touch upon deep learning and machine learning, right? So because I will be using that in these applications. Yeah. So the two big category of machine learning. So deep learning is a part of the machine learning. It's a new, very active category of machine learning. And two big areas of machine learning are supervised and unsupervised machine learning. So what I'm showing you here are some of the popular supervised deep learning models, including deep neural network, convolutional neural network, residual net or ResNet, and RNN, or recurrent neural network that includes LSTM, GRU. However, RNN is being replaced now by transformers, and then graph CNN. So supervised learning, as the name suggests, it's a learning process where the learners like us, or a machine that you're trained, right, is being presented with data, for example, in this case, images, and its true labels. Okay, so whether the image labels are whether the image is the type of animal that image is, right? Whether it's a bird, dog, and so on and so forth, right? So the goal of this learning is to build a AI machine to be able to predict the label of an image. When you present the image to the machine is being able to produce the labels, right? So the fact that it's a supervised learning is because this machine is during the training is presented with both image and its label, are both input and output of the machine. Okay, so the process of training involves presenting the machine with multiple pairs of these image and labels, or input and outputs. You have to present machine with multiple pairs. This is very much like a human learning, right? So when we teach kids about these images, you present an image to the kid and tell the kid that this is a dog, so that the kid remembers or recognize the features of a dog and remembers this is a dog. So next time we present a similar image of a dog, then the machine is able to pick up and recognize. So to a large degree, this is very much like a human learning. However, for human, you only need to present very few images, whereas for a current machine, you have to present lots and lots of these pair of images, okay? And the pair of image and its labels. So this process is called a supervised training. So from a mathematical perspective, the goal of a training is to determine these neural network architecture, and specifically the weights associated with the architecture, or the parameters of the architectures. Okay, so once we train the model in such a fashion, and then the model can be used for predicting the labels of a new image. Okay, so this is so-called supervised training. On the other hand, another big area of learning is called unsupervised deep learning. So for unsupervised deep learning, here we only present the machine with data or images, but the label. So there's no label associated with the images. So therefore, the goal of the machine is to really understand the image, to be able to learn the features of the image that characterize this image. However, we do not really know what this image is, right? So one very popular deep learning model or unsupervised deep learning model is called autoencoder. So autoencoder is this is autoencoder, right? So autoencoder is trained to be able to reconstruct the image itself. Okay, so the way how you construct autoencoder is by adding a so-called decoder on top of the encoder, and then you train encoder and decoder together to be able to reconstruct the input image. So once you train the encoder and the decoder, you throw away the decoder, keeping really only the encoder, and then the output of the encoder are considered as a codes that capture the features of the image. Okay, so I'll quickly give you an example here. So the autoencoder here is being presented or trained using these handwritten digits from 0 to 9. However, the autoencoder is not told which letter, what digits each one of the image is. So once you train the autoencoder in that fashion, you can take the code of the autoencoder, and then what you can do is you can visualize the codes. Here you see these clusters. Each dot is an image in this two-dimensional plane. You see clusters of the dots or clusters of images, and looking closely to these clusters, you realize that these clusters really these clusters include images come from have the same digits. So in that sense, these codes capture the characteristics of these images, because the images with similar features are not clustered together. Okay. All right. So using these autoencoders, using the decoder. So even though when our goal is to extract the so-called features and codes, we threw away the decoder. These decoders are still useful. One can feed this code into the decoder to be able to so-called reconstruct the code. So here's an example where the codes that extracted from this handwritten digits are used to reconstruct. So here you see the reconstructed images. All right, so now let me get into some of the applications. So this is one application where our goal is to really understand the characteristic in the so-called human gene expression. So what you're seeing here, the data includes the measurement of a mRNA, the amount of an mRNA of a human patients and cancer patients specifically. So as I mentioned to you, we have about 25,000 genes, and each gene would produce a specific mRNA. So in this particular measurements, we have gene expression of 25,000 genes for each cancer patients. So here we have totally, well, all of these 25,000 genes, we focus on only about 15,000 or 16,000 genes. And then we have the data, we have the measurements, these gene expression measurements of over 9,000 cancer patients from the TCGA database. Okay, so and then our goal is to really understand the characteristic of a gene expression, to be able to see what characterize how different cancers are characterized. So for that goal, we built a so-called autoencoder. However, this is just not a regular autoencoder. So the architecture is structured based on the biology and specific biological pathways, so that in addition to capture at the output layer the characteristic of these gene expressions, we are also able to understand the biology or the pathways that are unique to different cancers. Yeah. So here's one example where after we train such model, now you see that again, samples of the same cancer now grouped together, suggesting that these we call last layer, these codes capture the differences of different cancers. And then by interrogating these links from the trained model, we're able to elucidate some of the mechanisms underlying these different cancers. Okay, so such model that we built is we call it the gene set autoencoder, or GSEAE. So here is another project where the goal is to predict metastatic tumors using, again, this gene expression. Okay, so the overall goal is that once we measure the gene expression of a patient, we want to build a model that takes such gene expression and then being able to answer these questions, including whether this tumor is metastatic or not. Okay, so if yes, if the cancer is metastasized, and then we ask what is the primary type, because metastatic cancer is a stage where the cancer cell migrates from the primary cell locations to a different tissue. For example, the metastatic breast cancer cells now migrate to lung tissue. So that's a type of metastasis. So therefore, we want to predict if it is a metastatic cancer, then we want to predict what is the primary tissue type, right? So these are some of the goals. And to achieve that goal, we first built a deep learning model that, first of all, being able to predict, given the primary cancer, the non-metastasized cancer measurements, the gene expression measurements from non-metastasized cancer samples, we've been able to predict the specific type of primary cancer types, whether it's a breast or lung or other type of cancer types. Okay, so here are some of the deep learning models and CNN specifically convolution neural network models that we designed to be able to or design specifically to characterize the unique features of gene expression. And yeah, so here I show you the performances. So that's of these of the trained models. Okay, so in all cases, it provides fairly pretty good performances. Okay, so let me, and then once we are able to predict the primary cancer types, now we proceed to ask whether given a metastatic cancer, whether we are able to predict its cancer type. And for that goal, we also able to build a deep learning model, okay, so that able to predict 22 different type of metastatic cancer types. Okay. Well, in this case, we deploy a new type of learning. It's called transfer learning to really address the challenges of a small sample size in metastatic cancer types, where we take the model that we train from primary cancer types and we transfer it to the small samples of metastatic cancer types to enable the training. So through this transfer learning, as you can see, we were able to improve the performance in terms of accuracies from 67% to 93%. And then we went on to build a model to predict new cancer types from a single sample. So as you can see in most of the biomedical research, and certainly in this particular applications, collecting samples is a very expensive process. So therefore, well, TCGA it costs us $300 million for this TCGA project to collect about 10,000 samples from these 10,000 cancer patients. So as you can see from this example, it's a very expensive process to collect samples. So we don't imagine that such a big project of collecting huge number of samples will be able to support it constantly. In practice, what is more realistic is that there will be small number of samples from different cancer types that have been collected. So here the question is how are we able to build these machine learning models that can be able to predict new cancer types based on very few samples, right? So just yes, sorry about the noise here. Okay, all right. So to achieve this goal, we propose a model called cancer Siamese. So the cancer Siamese takes a single sample, we call query sample of a cancer patient gene expression, and then adds a database, for example, TCGA, of many tumors, okay, with a single or few samples. So what we imagine is in the future that there will be one or few samples are collected for different cancer types. So in a very fine classification, we can treat each individual patient as a specific type. Okay, so imagine now we have a collection of tumor samples, each characterized with its specificity here. So the question here is when you have a new sample, you ask what type of a sample what type of a cancer this sample is. So we built such model to be able to take this query and then compare with the samples in the database, and then ask which cancer type in the database that this query is similar to. Okay, so this is very much like this type of learning in machine learning is called few shot learning. It's very much like query and image in an image database with few images of each category. Okay, so for that purpose, this is the type of model that we built, and the model is able to tell you the similarities of the query model with each one of the tumor samples in the database. So here, what I'm showing you is the so-called few-shot learning performances, or one-shot learning. Actually, in the database, we have only one samples for each tumor. So here are the one-shot learning performances on predicting primary cancer and predicting metastatic cancer types. Okay, so all right, and in addition to these cancer diagnostic efforts, we also studied how we can use these AI models or deep learning models to be able to prioritize drugs. So in this particular case, once again, we look at a patient gene expression data and then build deep learning models to be able to predict the responses of a large collection of drugs, including 250 drugs. So to train such a model, we use a database that categorize the responses of 265 drugs in over 600 cancer cell lines, different type of cancer cells. Okay, and we're able to train such a model. I'll show you the model in the next slides. Okay, so actually, in this case, we use not only the gene expression data, we also use the mutation data of the patients. So we combine these two data types and build a deep learning model that's capable of producing the drug responses. Okay, so to train such a model, we use again a so-called transfer learning process that takes advantage of large collection of the TCGA data, because in the cell line we have only 600 samples, where TCGA has 9,000 samples, whereas TCGA sample doesn't have drug responses. So what we did here is we first train autoencoders without having, in this case, without the need to have so-called labels or drug responses. And then afterwards, we add a deep model on the encoders of each data type's mutation and expression to combine them together to predict drug responses. Here are the so-called heat maps of the drug responses of these 600 cells of the row data. So each row is a drug, and each column is a cell. And this is the predicted drug responses, the heat map of predicted drug responses. You can see they're very similar, suggesting the prediction is pretty well. And here is a quantitative measurements in terms of the mean square errors of the drug response, the prediction. Okay, so using this predicting model, we're able to predict the responses of these 265 drugs in TCGA cancer patients, where we are able to prioritize drugs, okay, for a large collection cancer types. Okay, and also, and some of them are validated by existing data, some of the drug responses. Okay. So let me see. Okay, so let me just in addition to that, let me quickly run through some of these slides, then I'll finish here. So in addition to that, we have also worked that on applying these deep learning models. Specifically, in this case, we use a so-called deconvolution neural network model to be able to pinpoint where the so-called transcription factor binding is. Transcription factor is a regular protein of transcription. So knowing where it binds is also a key aspect of this molecular research. So in addition to that, we are able to predict the so-called RNA methylation sites in the mRNAs, and then of a large collection of mRNA methylation sites, for example, using deep learning. Okay, well, here I'm just showing you a few examples in my own research of how these AI models or deep learning models specifically can be applied to address some of these biomedical questions. So now let me conclude by telling you where we are in terms of this AI biomedical research. So I formulated at the very beginning about these few aspects that I envision that AI should possess, right, in the future to be able to really become a artificial agent, to have that ability to assist the human in research and in clinical practices. However, we're not there yet. So AI, the current AI does not possess many aspects of its functions. So what is really missing right now? So what's really missing, here are some of the aspects that I think that's really missing. First of all, there's a lack of theoretical frameworks for AI systems. One of the key reasons is we still don't completely understand what our own intelligence is. So therefore, there's a lack of theories in guiding us in building these intelligent systems. Okay. Another aspect that's missing is, as you can see in the few examples I presented there, the models itself, even though we attempted to really incorporate some of the prior knowledges in the model, but the model in general does not have that ability to really incorporate the prior knowledges. So the type of prior knowledge I refer to is these large amount of accumulated knowledges by scientists over the last decades or hundreds of years on biology, on biomedical systems. Okay, such knowledge now is very difficult to be incorporated in the model to give the model an intelligence even before presenting the model with the training data. Okay. So another aspect that's missing is that once we train the model, this deep learning model is considered as a black box. However, in biomedical research, knowing how the model makes a decision and knowing potentially the mechanisms behind such a decision is crucial. Whereas the current model does not have that ability to be interpreted or to be understood. Okay, so building a so-called explainable deep learning AI model is one key research focus right now. Okay, so in addition to that, model can mostly perform classification, prediction. It cannot reason and create yet. While there's still a long way to go before the model can create, but building model that can reason is also one very active area of research. lastly, in biomedical research, as I alluded to, samples are small. Okay, so how we can, whereas most of these deep learning models, especially in computer vision, relies on large collection of the training samples. So how we can build intelligent models using small samples is a key requirement for biomedical research. I showed you one example that we built using the so-called one-shot learning model. Okay, but the more research is still needed in this particular area. So lastly, when we touch on human, security and ethics are key aspects. So research into building secure and ethical deep learning models are also highly needed in this area. Okay, all right. So that's pretty much conclude my talk, and I hope I give you an overview of why AI is important in biological research and how AI can potentially be used. I hope this can, attract some of your attentions and possibly attract some of you get into this particular research area, right? So with that, I can take some of your questions.
Thank you, Professor. You just blew my mind today.
Oh, thank you.
I didn't know we can apply AI technology to molecular biology. I thought they use it just to collect data and for other purposes, for medical sciences. But today, you honestly blew my mind.
Oh, thank you. Yes. So we really want to build the so-called AI system that I wouldn't say function completely like a human, but possess some of the intelligent aspect of human, so that it can assist us better in performing biomedical research and assist clinicians in this type of diagnostic practices and recommending drugs, therapeutic strategy, and so on and so forth. So this is really something that we are striving for right now in terms of our research. Anybody else? Feel free to ask me questions.